Welcome to the Cancer Neuroscience Symposium Conversation Series. I'm Monty Montano, Executive Editor of Wiley's Advanced Portfolio. We're excited to partner with MD Anderson and host this series to hear from key speakers featured in the February 2026 Cancer Neuroscience Symposium. Thank you for tuning in as we explore ideas to advance cancer neuroscience together. Delighted to meet you, Dida, and delighted to have this chat about the work you're doing and sort of a little bit about you, and as we build up to the Cancer Neuroscience 2026 Symposium next February. So we tend to like to start these with just getting to know you, a little bit about you, the person. So maybe you were reciting offline some Turkish poetry. So my guess is you spent some time in Turkey, in Istanbul, perhaps. And so maybe talk a little bit about you, the person, and how you arrived into the Nobel laureate lab of James Ellison, and how that feels.
Yeah, it's lovely meeting you, Monty, and chatting with you today. So I'm Turkish. I was born and raised in Turkey, and I have a bachelor's degree in biological science and bioengineering, another degree in chemistry. So me at 15 years old, starting college, thought I would be a bioengineer. And my plan was to uh, redesign how we do metal implants. I was somewhat obsessed with it, although I have no idea. There's no one in my family who ever has a metal implant, but I thought when I get old, I would need it, and current design sucks. The reason behind that was like most people are okay once they get like a knee replacement, hip replacement, but 15% of world population is actually allergic to metals. So for those people, those therapy options are not the one. And as I was going through my degree, I realized that every single idea I had in my head, it involved one way or the other a biomaterial pushed into a human being, but I had no idea about immunology. And it coincided with me towards the last few years of my bachelor's degree, stepping into an immunology lab for the first time. And I realized I have absolutely no idea what immunology was about. So I decided to have a PhD in immunology. That led me to UT Southwestern Medical Center, where I have a PhD in immunology. I joined to one of the few human immunology labs at the time, thinking I would be doing something related to human immunology. But as usual, there's a Spanish proverb that says, if you want to make God laugh, make plans. So my plans in human immunology took a really weird turn into mouse immunology upon an observation in the lab, which was in between two different CD8 populations in blood. There is a differential expression of beta-2 adrenergic receptors. And my advisor told me, hey, we started to see if ADRB2 has anything to do in T cells, but who knows, macrophages do express this. Do you want to take a look at it? And all of a sudden, I end up with a mouse immunology project in the human immunology lab.
And
And,
was that in Jim Allison's lab, or?
this was in David Farrar's lab when I was in my PhD.
I see.
And I was in a human immunology lab doing a mouse project, a T cell
Yeah.
lab doing macrophage project, and stepping my toes into neuroimmunology, which at the time was a field I would say not many people appreciated. I think since I started my PhD, field is growing beautifully. And my main observation, which became the focus of my thesis, was if macrophages, monocytes, dendritic cells are stimulated through beta-2 adrenergic receptors in the presence of toll-like receptors, they become anti-inflammatory. And we had a plethora of models like in colitis, infections, sepsis, but nothing in cancer. So, as a fresh graduate, my proposition to Dr. Ellison was, well, I discovered this immunosuppressive pathway, and in cancer there's immunosuppression, and anti-CTLA4, anti-PD-1 deals with one, but still most patients don't respond. What if it's neurotransmitters? So when I started in Dr. Ellison's lab in 2018, there was no paper to my knowledge that mentioned that there are neurotransmitters in tumors, which they should be. Every single tissue is innervated, but no one has ever formally looked at it. And that became kind of my project when I started my postdoc. And throughout time, it evolved into this novel population of immune cells, mostly T cells, in tumors, which express dopamine beta hydroxylase and can produce norepinephrine. So I have been studying this neurotransmitter producer immune cells for the majority of my postdoc, basically. But I started thinking I would be making biomaterials, and I end up in a cancer lab. I guess you go to where science takes you. That's the perfect.
Yeah, it's fascinating to sort of see the trajectory of your experiences in the lab, and this conference is really highlighting cancer interactions with the nervous system and nervous system interactions with the immune system, and this triad of interactions, and your focus on sort of neuroimmune interactions is just super, I think, appropriate and central to what we want to learn more about. So I'm just thinking more about what you were saying, with the adrenergic receptors on macrophages. So these are neurotransmitter receptors, right? Which means that the brain and neurons are communicating through small molecules with the immune system directly. And and that in some way there's suppression of that immune response. This is part of what you're looking at. And I guess it's in two flavors, right? It's the adaptive suppression and also innate suppression. So perhaps you could say a little bit about sort of the role of sort of this neuroimmune interaction for suppressing adaptive response and also for suppressing innate response,
So I think,
especially in the context of cancer where you really want immune response to be effective.
of course, I think the whole field is really fascinating. There are some real commonalities between most neurotransmitters' effect on the immune system, which, to my knowledge, every single paper that came out so far showed that neurotransmitter signaling suppresses immune function. There are a few exceptions, but almost all this way. For the case of adrenergic receptor signaling, we know from both in vitro studies early on and chronic infection or acute infection studies, when CD8 T cells sees norepinephrine, a few things happen to them. First of all, they do not proliferate as much,
Hmm.
which in the context of cancer would be a detrimental thing because you would want your antigen experienced T cells to proliferate and be able to cope. Second, they would reduce their production of TNF alpha, interferon gamma, granzyme B, so they are less cytotoxic, less effector like. And there is an upregulation of PD1, and PD1 is one of our beloved canonical anti-inhibitory receptors. So, in a way, for the T cells, mostly for CD8 T cells, seeing norepinephrine crumbles their effector function. On the other hand, for macrophages, it's a little bit more nuanced, and I think the pathway is more, I, how I should say it, the pathway can be both direct and indirect. So it can be indirect through cytokines. So if there is less of TNF alpha, less interferon gamma, macrophages will not be responding to these cytokines to gain more phagocytic functions. So they will phagocytose less. Same goes for dendritic cells. So that trickles down to antigen presentation, leading an impairment. Or in the case of direct signaling, since adrenergic receptor signaling suppress adrenergic receptor signaling induces IL-10, which is on itself an immunosuppressive cue. It can create further immunosuppression through IL-10's effect
Mhm.
on them and on their surrounding.
Mhm.
And that's excluding norepinephrine's effect on the vasculature, the
Mhm.
stroma, the tumor. So overall, I think each cell that you would look at in tumor microenvironment will be impacted. But of course, how they're impacted will be different, and it would all depend on spatially where these cells are. This is
I guess it's complicated, right? I mean,
complex.
you mentioned something, and I want to just get back to that. That you were talking about sort of mouse models and human models and back and forth. So maybe just sort of open that up a bit and sort of compare for us sort of the neurotransmitter mediated suppression of adaptive and immune response in a mouse model compared to a say a human model. If you were to take CD8 T cells and macrophages from humans and expose them to these, these immunosuppressive neurotransmitters, what would the dynamics look like in distinction from a similar type of experiment in mouse? And the reason I ask is because, uh, the mouse immune responses are not identical. There's a lot of similarities to what you would expect to see in humans, but they're not identical. Maybe there's a myeloid bias. This is your research. So sort of compare for us, and the sort of the expected response in terms of neurologically mediated suppression in mouse versus human.
I think it's really something we need to evaluate case by case.
Mm,
Um, for certain tumors, the tumor immune cell composition in mouse models and
mm,
humans may be alike.
mm,
But in certain cases, there's a myeloid skew, or these tumors become immune cold tumors in mice. So I would say if you're looking as a system, it's a case by case thing. But if you were to purify each population from either human or in mice and compare in a dish, so far, we were able to see T cells, B cells, macrophages, dendritic cells. They all resemble their behavior in isolation.
mm,
However, when we put all of these cells in a complex three-dimensional microenvironment, since the compositions are different, it's hard to predict, and we need to evaluate case by case. And I think one thing that we cancer immunologists tend to overlook, because it makes our life a bit easier, is in humans, cancer progression takes years.
mm,
However, our most of our models, if they are syngeneic tumor implantation, it takes a few weeks at best. And maybe the longest models, let's say KP mice, KPT mice, that should take a few months, but still nothing in the sense that human cancer progresses. So, irrespective, independent of the model that we are looking at, the human cancer and mouse cancer are inherently different just because of timeline, if nothing else.
mhm, mhm,
We also implant a bolus of cells, and they're all homogeneous, but in human cancer there's sequential mutations that result in microniches of different cancers within the same patient. So in a way it's impossible to truly model human cancer, but it's also impossible to modulate anti-tumor responses in a person just to test it out too. So I guess we need to know the weaknesses of our models and interpret our results best we can based on the weaknesses of the model.
mhm. I suppose they said at one point that it's impossible for humans to fly, and then we invented airplanes, right? So maybe the technology will help us in this. And I'm just sort of thinking about your background in bioengineering and biomaterials, and so my guess is you probably do think about the tumor microenvironment in terms of like the ECM and just sort of the structure of the whole immune infiltrate. And does that immune sort of micro infiltrate differ in mouse and human? And have you thought about looking at spatial transcriptomics and just sort of cataloging the different cell types in the context of the TME, given your background in sort of biomaterials and bioengineering?
We did not do extensive work, unfortunately. However, comparing at least the models that I'm working on to human transcriptome, existing human data sets on melanoma,
Mm,
the neurotransmitter expressing immune cells, for example, the focus of my research, we actually found out about these cells from a publicly available single cell melanoma data set.
mm,
And we were able to observe in that data set, and actually a few more others after that. In humans, it's mostly T cells that express DBH in tumors. However, in mouse, it's not just T cells, but your macrophages express it, your B cells express, and there's a
Mhm.
kinetic component to it. At a certain point, T cells lose the expression, for example. So even though there are some similarities to encourage a mouse study, as I mentioned, it's impossible to have at this point a head-to-head comparison.
Mhm.
However, you're right. There are certain techniques that are in development. Organoid cultures will hopefully be more dominant in the field, where we end up growing sections of patient biopsies. Maybe we would be able to recapitulate the tumor microenvironment. I mean, it's possible to envision an experimental system where we characterize, for example, melanoma ECM and use that as our basis to culture melanoma cells, immune cells, instead of a pure matrigel or collagen.
Yeah, these heterogeneous organoids, I think, are people are beginning to try and use them.
I think so too. I think right now the biggest problem with that aspect of the field is it's really cost prohibitive,
Mm.
and you can't do large scale experiments with those because, first of all, if you're dealing with patient samples, unless you are a lucky person who got a huge chunk of tumor, your tumor sample is really small.
Mhm.
If you're reconstituting the tumor microenvironment, there is still the problem of all these cells will reorganize artificially, which will not be able to recapitulate what
Mhm.
we see in human biopsies or in mouse tissues, which develop more naturally. So again, each system has its weaknesses. I keep
Yeah,
telling, but
and I think it speaks to this larger issue of creating teams that approach a particular kind of cancer set of questions from different perspectives, and it just further makes the case that having scientists with different backgrounds, having scientists from different perspectives, who have different life histories and sort of scientific lenses, if you will, can make the observations that more robust. So I wanted to maybe switch gears a little bit and ask you, because I know you've been doing some work, and obviously with Jim Allison on immunotherapy, and thinking about how to better implement and leverage, the immune response to limit cancer metastasis. And, in the real world, people often have inflammation. They have various other morbidities. And so, could you speak to the aggravating role of inflammation in immunotherapy and the type of work that you've been doing to sort of suppress uh, or to modulate that aspect of inflammation aggravated immunotherapy?
right. So for my even the word inflammation, I'll probably be stuck in the linguistic throughout our podcast recording. Um, the word inflammation is a single word that encompasses so many biological phenomena, right? The inflammation might be coming from a high-fat diet-induced obesity.
Mm.
It might be a inflammation that's locally there because there's a minor cut and there's some local immune responses. It can be someone is going through a vaccination process. So each of these cases are things we can model in mouse clearly. And in humans, I think our biggest hope to answer these correlative observations would be studies like NHS's biobank, where they have hundreds of thousands of individuals with a detailed health recording with who is getting whatever medication, what disease they have. For my research, my primary observation, which, as I mentioned, is the neurotransmitter producing immune cells. These cells, interestingly, appear more in animals that are treated with anti-CTLA4. So, in one way, anti-CTLA4 therapy is alleviating an immunosuppressive pathway
Mm.
and giving T cells a chance to be more active. And even ipilimumab alone has changed the clinical outcome drastically. However, in mouse models, and we were able to verify this using multiple mouse models in really detailed kinetic studies. When animals see anti-CTLA4 or anti-CTLA4 and PD-1 together, we observe a robust increase in DBH expressing T cells. So while we are alleviating one immunosuppressive pathway, this signaling results in production of norepinephrine producer T cells themselves would be immunosuppressive on their own right. So my proposition towards the end of my postdoc was maybe we should combine DBH inhibitors with immunotherapy to give immunotherapy a higher chance to be successful. And of course, DBH inhibitors are not FDA approved yet. There are some clinical trials for example, PTSD, which it will be a tangent, but most people don't think psychological disorders is an immunological one. But there is vast literature on how psychological disorders have an immunologic component, where patients, for example, with depression have a disbalance in.
Well, depression has been linked to elevated levels of stress hormones and inflammatory factors like TNF and IL-6 that are CRP. So I can see a connection there.
Yeah, I mean, you're in the field, so, but when I talk about these non-immunologists or non-neuroimmunologists, they're like, hold on, that's too far-fetched.
Yeah, yeah.
I think the blessing and the curse of this particular field is you need to learn a lot, read a lot, and a little bit dream outside the box, because the field is not, you cannot solve a problem with your neuroscience skills alone or immunological skills alone. You need to be able to shuffle different hats and maybe wear a few different hats at the same time. But it's a fun place to be as a young scientist, because
Hmm.
ample opportunity to carve your own niche in a way.
Yeah, I mean, I think you're emphasizing the need for multiple hats in the room is absolutely the way science is moving forward nowadays with writing, and I'm sure you're writing K99 or KO1, or you're involved in writing grants. That more grants these days require, assuming that the funding infrastructure continues going forward, which we hope that it does. Most grants involve team approaches, and when the NIH review committees, and I've been on those committees, look at these, they're often looking for a robust set of individuals who can see the elephant from different perspectives, as it were, the old metaphor. But I wanted to turn back to the this idea of suppressing the immunosuppressive, activity, and how that is beneficial. Obviously inhibiting checkpoint inhibitors can, relieve the blockade in T cell function and T cell, management of a tumor. But that excessive activity may itself be a problem. I think you were sort of alluding to this. And so, it represents a sort of a fine tuning, if you will, that you need to have checkpoint inhibitors so that you can liberate immune response to fight tumors. But that liberated immune response checkpoint inhibitors were created presumably by evolution to limit immune response so it doesn't damage everything. But if you don't create boundary conditions, you may have other kinds of adverse events, like I think even you have published on myocarditis and maybe even fibrosis. So could you speak to sort of this sort of highly sort of tailored approach that we need to take to both liberate but also modulate immune response so that it's fine tuned, and how the sort of challenges that raises for the field.
Yeah, of course. I think you're raising a great point, because in on paper, all of these ideas work greatly. Let's alleviate all suppression from T cells, but then you result with plethora of immune-related adverse events, from colitis to myocarditis. and the results can be quite severe, where patients may take lifelong medications to deal with these IREs, even though they are now cancer free. So I think in our, for example, myocarditis study that you mentioned, we proposed the use of CTLA-4 Ig, which we show that impacting by CD80-86 pathway can be still effective in anti-tumor responses, but in this model, we don't observe much myocarditis. The problem with, I think, immunology in general, and I love criticizing my own field, for us, it's such a large network of relationships through cytokine networks, through neurotransmitter networks, different cells interacting, and we tend to think these systems in isolation because it's easier for human brain to conceptualize. Your
Hmm.
cell type is impacted by cytokine of your choosing.
Right.
In reality, of course, it's much more complex than that. So I think cases like IREs in the case of cancer, or if you're stepping one step and looking beyond cancer, in neurotransmitters in management of psychiatric diseases, in asthma, in skin conditions, we need to be more aware of the system that we are dealing with to understand what are the possible tipping points, sort of, that
Hmm.
we envision. Going back to my own work, with we did not know that CTLA-4 treatment would result in yet another immunosuppressive pathway. Now, in animal models, when we alleviate the second pathway, now we add DBH inhibitors and result in better anti-tumor responses. At this point, I think we did a fair job, but I'm pretty sure someone else will do an even more in-depth analysis and say these two treatments trigger a yet third immunosuppressive pathway that we need to deal with. I think we need to go back to basic science and try to understand what the impacts of these combinatorial treatments that we are doing
Mm.
on a cell basis, as well as to model as much as possible to understand the general implications of our treatments so we can foresee what are the potential adverse events. But going back to our earlier conversation, mouse and human are not the same. So these IREs that we observe in patients, unless you have specific transgenic animals, these are most of the time are not observable in mice.
Mm.
So from a mouse immunologist standpoint, the animal looks perfectly fine, and their tumors are cured. You trigger a potent immune response, and your urge is to move forward. However, due to inherited differences of the systems, some of these treatments may trigger adverse events in humans. So I think my short answer to your question is, as we understand the systems more, we would have a better understanding of what would be possible adverse events and maybe foresee it. And for that, maybe small scale clinical trials would be a good place to start.
Hmm. Yeah, I mean, I think that absolutely. I mean, small scale clinical trials are closer to the beast, if you will, if your focus is on alleviating human cancers. But I think you're really onto something here. I think the idea of integrating and understanding the interactions not only between organs but also different chemotherapeutics and drug regimens is spot on. And you know, you mentioned some of these adverse events associated with immunotherapy and chemotherapy, and I think you mentioned colitis, and it made me think about, you know, this emerging field of the gut brain axis. You know, and I just wonder what your thoughts are on that, because we know that, for example, that somatosensory neurons innervate the gut. We know that bacteria, the microbiome, produce neurotransmitters, dopamine, glutamate, you know, norepinephrine, and we know that there's feedback between the microbia in the gut and the brain. And so how, I wonder, you know, when just we think larger, picture than just maybe just the TME. But the larger picture of the human being or even the mouse model where the gut, I wonder, could you speculate on whether that's something given the emerging field of the gut-brain axis?
Yeah, I think that's a fascinating field within both neuroimmunology as well as intestinal immunology in general. So even this field of gut microbiota is so context dependent, right? People who are consuming Western diets or their traditional
Mm.
diet will have different microbiota, even though they are genetically similar. I think there was a cell study a few years back that showed people who migrated from, if I recall correctly, Tonga or maybe New Zealand, somewhere. I think it was one of the micronations. And once they encounter Western diets, their whole microbiota, whole physiology changes. And I don't think the changes are for the better, but uh, it, it's it's possible to mimic some of these work in animals to have better understanding of how gut microbiota impact tumor responses and impact nervous system and contribute to the milieu. But I think again that goes back to finding different cohorts of patients who, for example, are treatment naive, who started their treatment and they are responding, who got a secondary line of treatment. And these are, compared to everything that we've been discussing, is I think the most modelable version of tumor immunology. I think the field that you mentioned is fascinating because intestinal system is also, even though it's called the second brain, it's also the largest immune organ.
Mm.
There are more immune cells in intestines than anywhere in your body. And there has been a few beautiful studies, mostly from Daniel Nusida in Rockefeller, where he has shown this intricate relationship of adrenergic receptors and intestinal macrophages, and
Mm.
how even being close to macrophages
Mm.
changes their morphology as well as function. So it's a really layered work, I would say, if you want to study the effect of gut microbiota in the context of immune-related adverse events in patients through the lens of even this is a long sentence, but I think the whole field so far, we there has been several constant observations. I think we can take them almost true at this point. For example, we know that being away from homeostasis changes gut microbiota, whether that is a psychological stressor, a physiological stressor like cancer or an infection, gut microbiota is bound to change. How it changes depends on where the individual is, what kind of external bacteria, viruses, archaea, fungi that they are being exposed to. But there will
Mm.
be a change. I think if and at this point, I want to take my dream big head and I want to introduce probably AI for this. If we were to analyze, I think we're at the point where one person looks into data and makes an observation phase of science has passed. And I think we are beyond the phase someone programs and comes up with a few intelligent hypotheses. What I see the field, where I see the field is moving is maybe we, as scientists, collect as many different data sets as possible. We get
Mhm.
samples from our patients. We get biopsy,
Multimodal sampling. Yeah.
and then we feed them into maybe an LLM and let the artificial intelligence to look patterns that are beyond our comprehension
Hmm.
and do this in iterative format where once we get more hypotheses to test, we feed this in a more active learning fashion back to our system to come with hypotheses that we wouldn't be able to see otherwise.
Hmm.
That somewhat diminishes the role of scientists, maybe, because we are always the ones who come up with the hypotheses.
Oh, I don't know. Actually, I think that it's a fascinating this supposition that you could collect lots of different types of data and then put it into a machine learning algorithm, an AI algorithm, and come out with patterns or predictions based on some subset of that input data. And I mean, it comes back to something that I think you were mentioning earlier. People, the differences in response to treatments. Some people fare well, some people not so much. And we saw that with COVID, right? The people that were on IL-6 inhibitors, tocilizumab, some did really well, and some were non-responsive. And identifying responders versus non-responders will in part, I think, no doubt benefit from machine learning cluster identification, where you have clusters of people that are resilient, clusters of people that are high risk. But then you have to bring the scientists back in to say, well, okay, let's look at what what were the driver data sets. Were they gut? Were they stress? Were they exposome? Were they genetics? And iterate. And so I think there's going to be possibly this partnership between AI and the biologist to continue sort of this iterative process of learning and redirecting the search towards our goals, which is to improve the human condition. So are you starting to sort of build these AI models and machine learning models? And
Unfortunately, I'm not really savvy in programming, but I have two amazing collaborators who are both computational biologists.
excellent.
One of them happens to be also my husband, so that's really handy.
So the
If
work never
I
stops. I can see the second part of your work day begins when you get home.
Yes, exactly. And his workday never ends because I always keep asking him more things. But going back to your point in team science and now coming into more computational aspects of the field, I was fortunate enough to collaborate with two computational biologists because I was collecting all these imaging data sets, single cell RNA-seq data sets. And I would look at these files and like, cool images,
Mm.
lots of Excel numbers, and I have no idea how to deal with this. Definitely having someone on the team that understands the biology helps. And as time progresses, I think even my experience with computational biologists has changed. When I started as a graduate student, there wasn't much of computational biologists. There were computer scientists starting to do computational biology, and they didn't understand what we were talking about. Now, with their integrative training, there's more of them that understand. But I think we ourselves, within our group, we don't do LLMs. However, there are a few interesting examples of LLMs integration in biomedical data. There is, for example, Gary Nolan's efforts from Stanford. His take is a little bit different. He is integrating single-cell RNA-seq data with mass cytometer data to be able to link protein expression into transcriptomic data to find these cells that he's only observing with 40, 45 markers to predict what their transcriptomic data would look.
I did my PhD with Gary Nolan.
I did not know of that, really. Yeah, that's really cool. I mean,
I was like, wow, you really do your research. Yeah, Gary's a good friend as well.
Gary is a fantastic researcher. Also, he is one of the faculty members in the Ellison Institute. So we had some opportunities
But
to.
he's at Stanford still. I mean, you're gonna have a hard time getting him to go to
He
Texas.
sometimes flies to Houston. I don't know
Does
whether
he? Yeah, that's good.
whenever
Yeah,
he wants brisk. I don't know what his frequency is,
he
but
might use the spaceship since he has a lot of friends up there.
it might be that too. Um, but in our conversations, when he explained this idea, I thought it was really fascinating because mass cytometry is much more accessible than doing large scale transcriptomic analyses. And being able to predict, for example, transcriptomic data would open so many experimental questions. Also, I think from my standpoint, I wasn't as I had nothing to do with cancer when I was in my PhD training. I was thinking I would be doing human immunology from the lens of infection and inflammation.
Mm.
And as I was going through my classmates' talks in cancer biology programs, they were all talking about five or six proteins, KRAS, P53, P10. I mean, one can count them at one breath. And I was like, there are 20,000 genes in human genome, and we are stuck with five of them. Come on, we can do better. And as I propose LLMs, I think something like an LLM would be able to find out a paper that was published in a journal that I would normally read and say, there's this link that's potentially important for this perspective. That's the link I'm expecting LLMs to make. And of course, this hasn't happened yet, but the field is in its infancy. And as we develop more tools and collect more data, I would envision that LLMs will be part of our daily life as scientists.
Mm. I think we probably have to finish up, but before we do, could you, Dita, just sort of talk? I mean, you're such the perfect example of the sort of an adventurous spirit in science, and just crossing different disciplines. And can you provide some advice for like grad students, international grad students from California, Boston, just people from different backgrounds? What advice do you have for them to, when they start in one very siloed field, but what motivates them is maybe perhaps something much bigger, but and are reluctant to cross those barriers? What would you say to them to get them to consider that?
I think I have a few observations from my own experience that will hopefully be helpful for future generation of scientists. So, of course, we're all inherently curious, otherwise we wouldn't be scientists. This would be a horrible field if you're not curious. But assuming that you are curious, you don't need to be curious about the project assigned to you. I'm sure it's impactful. I'm sure it's going to change the field somehow. My thinking was, as a grad student, I'm expected to learn. That's the only thing I'm expected to do, and do some decent science on the side that leads to publications, and hopefully my PI gets a grant. So to that end, I was going to seminars after seminars with the UT Southwestern, and I was lucky because we had a really beautiful biochemistry department, an incredible neuroscience department. For the first few years, I had little understanding of what they were talking about. I mean, there was free food, yay for grad students. But as years passed by, I realized that I understood more and more. And even to this date, I can bring in models from different fields to my own work that a regular immunologist is not even aware of. So my first recommendation would be go through seminars, force yourself to read papers. On paper side, I can read excessive number of papers in a non-humane fashion. My recommendation to all grad students in my PhD program as well as my postdoc program was have a system to read papers. So for me, Mondays, the first few hours are dedicated for papers, and I don't sit and read a paper. The way I do is I have a folder of all the journals I like. So there are at this point 35, 36 journals where I bookmark table of contents. Every Monday, all of those tabs open. For
Hmm.
journals that are monthly published, you have already seen the table of contents.
Hmm.
I read through the lines, and even one line is a punchline of the paper, tells you a lot. You learn what's new in the field, you learn who discovered it.
Hmm.
And if there's something of interest, I keep that paper. I open it as another tab, and in between centrifugations, I read an abstract. So with an abstract, you learn more than the title. And if something related to my research, I keep it. If not, I learn something. So I just close the tab. So I have this huge list of tabs that I keep closing. If there's something that passed the abstract stage, I look through the figures. You learn a lot from by looking at figures. A lot of papers get eliminated again. And then there's one or two papers every week that I think I should truly read. And those papers come home with me on my tablet or print out. So I actually read those papers. But even though I didn't read papers in most people's sense, I learned quite a lot by skimming through literature. So my recommendation to people who are in training is read. I mean, that's the only thing you're paid to do. You are supposed to learn. Otherwise, it's hard to find these commonalities between different systems, different disease models, or come up with ideas that seem out of the box. I mean, if you don't know what outside the box looks like, how are you going to come up with ideas outside the box? So those two would be my two recommendations. Go for freelance seminars and read papers. But
Excellent. Excellent. Well, I think we can maybe stop there. I mean, it's just wonderful to chat with you, Dita, and I wish you the best of luck. And we very much look forward to seeing you in Texas in February for this conference.
thank you so much for having me, Monty. Look forward to the symposium.
Yep. Thanks for tuning in today. We hope you'll join us as we continue the conversation in Houston and virtually at the 2026 Cancer Neuroscience Symposium hosted by MD Anderson and Wiley's Advanced Portfolio.